So
you can take off your mask if you want. Your mask.
Oh,
Yep.
sorry.
You're good.
Okay.
Just so I can hear you better.
Exactly. And start. So, good morning, everyone. So, welcome again to this lecture. So, I just have realized, or I just have the impression that I yesterday did a lot of stuff at a time. So, it will be too much for some of the students. So, I just want to make a pinpoint of important issues what we have discussed yesterday, especially things related to the gametogenesis and the ones which are related to sex determination. So in terms of gametogenesis, a very important aspect of gametogenesis is that it happens in the male gonad, that is the testis, and the female gonad, the ovary. I'm writing it down because it is much more easier for you to follow so that you can understand the important aspects of gametogenesis. So in both cases, it starts with mitosis. Just mitosis is a duplication of the cells without any division. That means following after DNA replication. So the daughter cells which are resulting from mitosis are exactly the same as the ones from which they arise from. That means these cells are totally different, it's the same as the other cells in terms of mitosis. So mitosis happens in the primordial germ cells, and this gives rise to primary spermatocytes. Primary spermatocytes in the testis, and the same thing here, that the germ cells are dividing or mitotically dividing, and this gives rise to the primary oocytes. This is the main difference now. So this is the process of primary spermatocytes will be further going into the process of the first meiotic division, meiosis 1, which gives rise to the secondary spermatocytes. Normally these ones are 2N, that is diploid, and these ones are only 1N. In this case, after the first meiotic, give to secondary oocytes. So then further, subsequent second meiotic division happens, and the secondary spermatocyte develop into spermatids, which are also 1N, but they have a single chromatids. The other one, the secondary spermatocytes are undergoing the second meiotic division, meiosis 2, during the time of fertilization. So here we see that, so this whole process in the testis happens throughout the life of an individual after puberty. That means after puberty in the male testis, mitosis and subsequent 2 cycles of meiosis are happening to get rise to, after spermatids are formed, there is a differentiation of these spermatids to give rise to sperm cells. So this process, the whole process of mitosis and also meiosis of the germ cells happening after puberty, that will be a continuous process in the case of males. But the case in female is different, in which that at birth the female have a specific number of primary spermatocytes which enter into the phase of meiosis 1. That means there will be a first meiotic arrest here. So after meiotic arrest, the oocytes, or the primary oocytes, are at the meiotic arrest of 1 at the time of the birth of the baby girl. So at the time of birth of the baby girl, these spermatocytes are around 2 to 4 millions in number. However, during the process, or from the childhood to puberty, at puberty time, when the ovaries are fully functional, then these meiotically arrested oocytes will develop into this, will finish their meiotic divisions and give rise to the secondary oocyte. These secondary oocytes are the ones which are ovulating and ready for fertilization. That means that from here to here at puberty, the number of primary oocytes at the time of puberty range between 300 to 400,000. So this shows that a significant number of primary oocytes are deteriorated or degenerated from the time of infancy to the time of puberty. That means at the time of puberty, a baby girl, also a girl, has around 400,000 of primary oocytes which are ready for further divisions using meiosis. So these secondary oocytes, which are the results of the first meiotic division or ovulating, are going to the oviduct, where fertilization takes place. When the sperm and the egg are united, then before the unification of the maternal and the paternal genome, so there will be the second meiotic division, which gives rise to a fertilized zygote and the secondary polar body, which is the smaller portion of the oocyte which will be degenerated and it's no more functional. So this is the main differences, that the baby girl at birth have millions of primary oocytes which have already entered into the first phase of meiotic. So they are arrested at that stage, and they will continue to, they will start their meiotic division at the time of puberty, giving rise to secondary oocytes, which is the one ovulating, and these ovulated oocytes will go to the oviduct. At the time of fertilization, then the second meiotic division happens, and this polar body will be released, and the fertilized zygote will be a diploid one. That means that this will be to N. That is N. So that is the reason why we have in males a continuous supply of primordial germ cells from mitosis and subsequent meiosis. So this is the process which happens throughout the life of the males. However, the females at puberty starts with 400,000 of primary oocytes, which will be cycled and utilized in every cycle, every month after puberty. There will be 10 to 25 primary oocytes which will be selected. Out of these ones, the majority of them are degenerated, but only one will be ovulating after reaching to the secondary oocyte. That's why, so this is the amount of the number of primary oocytes or secondary oocytes, which is primary oocytes, which is available at the time of puberty, and this will be leading to, so there will be no additional germ cell multiplication through mitosis after the baby is already born. So that is the difference between the gametogenesis in the testis and in the ovary between the male and female. So I have tried to show yesterday there are two events. There are two events in the meiosis 1 which are responsible for having different genetic combination of sperm and eggs. The first one is which I called the crossing over. This is a crossing over is the exchange of segments of chromosomes. That means homologous chromosomes between two non-sister chromatids. So that is the first place where crossing over is contributing to genetic diversity, what we are observing during the process of gametogenesis. The other one is the random distribution. Random distribution of maternal and paternal chromosomes. So paternal, not chromosomes, but chromatids of the homologous chromosomes. Paternal chromatids during the time of the first meiotic division in the secondary spermatocytes or in the secondary oocytes. So these are the two main events which are happening in the time of the first meiotic division. In the process of meiosis, there is a crossing over between segments of the two non-sister chromatids of the homologous chromosome. What we call is homologous gene combination or recombination. This is called crossing over. The other one is the random distribution of those maternal and paternal chromatids to the daughter cells, which are the so-called secondary spermatocytes and secondary oocytes. So these are the two important aspects what I have discussed yesterday. Today, so as I said before, that through the combination of the sperm and the egg, there will be a new individual form. It is called the zygote at the time of fertilization. So the question here is, what is the sex of this individual which arises from the fertilization of the sperm with the egg? So that is the so-called sex determination, what we have talked yesterday. So sex determination so the establishment of the main reproductive organs, either it is a gonad, internal reproductive tract, or external genitalia, is dependent on the sex determination. Sex determination can happen at 3 different levels. The first one, which is very important one, is the chromosomal sex determination. The chromosomal sex determination, which is called a genetic sex determination, this is it normally occurs at the time point of fertilization. It is fixed, I mean the chromosomal sex of an individual is fixed at the time point of fertilization, and it depends on the inheritance of inheritance of either X or Y chromosome from their parents. A male individual, or the paternal genome, contains 2 types of chromosomes, sex chromosomes. The first one is the X chromosome. Either they can contribute either the X chromosome, or they can contribute the Y chromosomes. The maternal side have only X chromosomes. That means X. That means any oocytes which is developed from the mother contains only 1 X chromosome. However, the sperm which is produced by the male can have either X chromosome or Y chromosome. Depending on which chromosome the individual inherits during the time points of fertilization is the one which determines the chromosomal sex of an individual. If an individual is getting 2 of the X's, then it will be XX, then it will be a female. If an individual is taking the Y chromosome from the father and the X chromosome from the mother, then that individual is a chromosomal sex of XY. Then it will be a male. So this shows that the chromosomal sex is simply the combination of sex chromosomes of an individual, which is dependent on its inheritance from the paternal side or from the maternal side. So this is the very important step of sex determination, which determines also the next step of sex determination, that is the primary sex determination. We will come to that. So the second phase of sexual determination is the primary sex determination. Or what is called gonadal sex determination. So the gonadal sex determination is normally occurs following chromosomal sex determination. Chromosomal sex determination, that is whether an individual is XX or XY. So this is a development. The primary sexual sex determination is the development of a male or female gonads from undifferentiated bipotential gonad. That means if an individual or an XX fetus is a female, or when an XY fetus is or a male. So these two combinations are determining which type of gonads will be developing in the fetus. For example, if in the XX fetus, the very important gene which is called in XX fetus, the very important gene which is called R-spondin, RSPO1, RSPO1 gene is a autosomal gene which is located in chromosome 1 of the genome, of the human genome, and this will be activated in the absence of the Y chromosomes. In the presence of the Y chromosomes in the male, what is important here is that the male Y chromosome contains a gene called SRY gene. This SRY gene is that the sex-determining region of Y chromosomes, it's called, is important and giving rise to, this is coding for the SR1, SRY protein, which is a transcription factor activating the SOX9 gene from the autosomal gene region. So this SOX1 gene, SOX9 gene, which is activated here, have the potential to inhibit the activity of the signaling pathway, which is activated by the XX chromosome. That's, for example, RSPO1 is activating the gene signaling pathway called the Wnt4 and beta-catenin pathway. So this beta-catenin in with the Wnt4 and beta-catenin pathways is the one which is supporting the development of the ovary when the fetus has an XX chromosomal combination. So this will give rise to the ovary, the development of the gonad ovary. And in this case, the SOX9, which is also activating the signal cascade, which is supporting the development of the testes. These two processes are antagonistic to each other. That means when in the presence of SRY gene, when the fetus is XY, then the SRY gene will code for a protein called SRY protein, which activates the SOX9. The SOX9 is inhibiting this signaling pathway to happen. That's why there will be no ovary. The cascade will be developing to the testes. However, when the SRY gene is not present, that means when the fetus is XX, then the RSPO1 is activated as a default program, and this will lead to the activation of Wnt4 and beta-catenin. This pathway inhibits the activity of this SOX9 gene, which is an autosomal gene. So the activity of the SOX9 gene will not be activated because of the absence of a SRY gene here, and at the same time it will be also inhibited by the activity of RSPO1 and Wnt4 and beta-catenin pathway, so that there will be no testis development, there will be only the ovary development. This is the molecular pathway in which gonadal sex differentiation happens, either in XX fetuses or in XY fetuses of the male. So undifferentiated fetal gonads contains a kind of structure like this. There are different types of ducts here. That is the gonad, which can develop to either to the ovary or to testes. So this gonadal tissue contains primarily 3 different types of cell types. One is the somatic support cells. The other ones are the precursor steroidogenic cells. These are the cells which have the ability to synthesize or to release steroids. Or the third ones are the primordial germ cells. So depending on the how the gonad is differentiating either to the ovary or to the testis, these 3 types of cells will be differentiating into different types of cells. The somatic support cells, for example, when the gonad is becoming, that is in XX fetuses, that is in XY fetuses. In the ovary, when the ovary is developing from the undifferentiated gonad, then the somatic cell support will be granulosa cells. And the precursor steroidogenic cells will be the TECA cells. How these different types of cells in the ovary looks like, we will see in our class, which is maybe next Monday, when we are talking about the female reproduction system. So the TECA cells, and the other ones are the primordial germ cells, are the oogonia cells. So if the fetus is XY fetus, then it develops to the primordial or the undifferentiated gonad will develop to the testis, and this testis, in the testis, the somatic support cells will be developing to Sertoli cells, which are important for the maturation of spermatozoans, and the precursor steroidogenic cells are developing to the Leydig cells, which are very important cells of producing testosterone in the testis, and the primordial germ cells are developing to spermatozoas or spermatogonial cells. Spermatogonial cells, which will be used for further mitosis, meiosis, as we have seen at the beginning. So this is the very important aspect, how gonadal differentiation is the basics for differentiation of different types of cells in the undifferentiated gonad to different types of cells, which are important or playing a role in producing sex steroids in the gonads, and also they are important for the spermatogenesis or in the process of oogenesis in the ovary. So what we have seen so far is what will happen here in the undifferentiated gonad structure, but there are a lot of different types of ducts here that is called the mesonephrons. Mesonephrons. The mesonephros is the one which is attached to to the differentiated ovary or testis, fetal testis, and this part of the gonad is having different types of ducts. As you can see here, that the genital ridge, which is developing either to the ovary or to the testis, is in the middle, and the blue one here is showing that is the Wolffian duct, and the Mullerian duct is shown here as a red. In the presence of the SRY gene, or in the presence of a testis, testosterone and anti-Mullerian hormone are the two hormones which are important in the differentiation of this process. That means that the testosterone, which is produced by, let me write down. So we are already now in the third type of sexual determination or sex determination, that is the phenotypic or secondary sex determination. So phenotypic or sex determination or secondary sex determination is starting to work after the gonads are differentiated. The gonads are differentiated either into fetal ovary or fetal testis. And then it is normally the differentiation of the female or male the differentiation of the female or male internal reproductive tracts. So this is dependent only on two main hormones. One is testosterone, which is produced by the Leydig cells in the testis, in the fetal testis. The other one is the anti-Mullerian hormone, or Mullerian duct inhibiting substance, it's called also anti-Mullerian hormone. Which is produced by the Sertoli cells in the testis. So what are these two hormones are doing during the process of phenotypic sex determination? The presence of testosterone in the testis gives rise to the Wolffian duct to differentiate into the different parts of the internal genital or internal reproductive tract in the male. That is, so the testosterone is giving or initiates the Mullerian duct, the Wolffian duct, sorry. The Wolffian duct to differentiate into epididymis, the vas deferens, and the seminal vesicle. These are very important internal reproductive tract in the male, which are playing a role during the process of spermatogenesis and also the maturation of the sperm. So the epididymis, which is attached to the testis, the vas deferens, which is a tube-like structure, and the seminal vesicle is the vesicle which is producing most of the fluid in the semen, in the fluid in which the sperm is swimming in, is produced by the seminal vesicles. These are the three types of internal reproductive tract components in the male which are arising from the Wolffian duct. What happened to the Mullerian duct? The Mullerian duct will be forced to degenerate because of the anti-Mullerian hormone, which is synthesized by the Sertoli cells here. So the anti-Mullerian hormone forced to degenerate the Mullerian duct so that there will be no female reproductive tract development. However, when we have a female testis, a female fetus which have ovary, then these two hormones are no more available. So if these two hormones are not available, that means that if there is no testosterone, the Wolffian duct will not go to differentiate to the different parts of the male internal reproductive tract. And because of the absence of anti-Mullerian hormone in the ovary, so the degeneration will be stopped. That means that the Mullerian duct will develop to different types of the female reproductive tract, that is the oviduct, the fallopian tube, the uterus, and some portion of the vagina. So this is the way how it works. In case of the presence of testosterone and an anti-Mullerian hormone, which are synthesized from, which are released from the Leydig cells in the testis or the Sertoli cells in the testis, the Wolffian duct will remain and develop into epididymis, vas deferens, and seminal vesicles. However, in the absence of these two male hormones, then the development of the Mullerian duct will be initiated. That will end up to the oviduct, uterus, and the vagina, as you can see here. So these are the two pathways in which the secondary structure, the secondary sex determination happens, in which depending on the gonads which is present, then the development of the different types of reproductive tract will be initiated. So just to give you an idea, is relatively to understand it, so a colorful one, so that you can understand what it means. Here we have 5 to 6 weeks of embryo, human embryo, which have both and different types of ducts, and the gonadal ridge is the one which becomes, the blue one is the gonadal ridge, which can become testis or ovary, depending on the chromosomal sex differentiation at the beginning. And this is the position of the kidneys, just to show you the relative position of the different organs. So in the presence of the Y chromosome, that means in case of males and XY chromosomes, then the pathway which is developing is different than in the absence of the Y chromosomes. That means in the case of females. We see here that we have the Mullerian duct is the one which is in purple, and the light green one is the Wolffian duct. So we will follow these colors and to show you how this development takes place in the process of pregnancy. You can see here at the age of 7 weeks of pregnancy that in the male, the Mullerian duct starts to degenerate, as you can see here, and the Wolffian duct becomes stable so that it will give rise to the different parts of the male reproductive tract, including epididymis, that is the testis here, the first difference, which is transporting the sperm cells to the seminal vesicles, where the sperm will be diluted with fluids, which are secreted by the seminal vesicles. Here, the urinary blood, and this shows that anatomically, the male reproductive system is combined with the urinary system here. So as you can see here. So that means the sperm is go out here through here. That means that's urethra. So at birth, we will have a male reproductive tract which arises from the Wolffian duct in the presence of the Y chromosome. In the other case, in the absence of the Y chromosomes, as you can see here, the Mullerian duct is, the Wolffian duct is degenerating, the light blue one is degenerating, and the persistence development of the Wolffian duct give rise, the Mullerian duct will give rise to the different parts of the female reproductive tract, including that is the uterine tube, that is the so-called oviduct, and the other one is the so-called uterus here, and some parts of the vagina are also here. As you can see here, the urinary bladder is here, that the female reproductive tract is relatively independent of the urinary system, as you can see here. So this is just to show you how the different duct systems which were present in the different stages of embryonic development or fetal development are taking the direction of the secondary phenotypic development, either in the male direction or in the female directions. As it has been seen also in the fetal gonads, so initially the development of the different parts of external genitalia are also derived from a common bipotential tissue. So this bipotential tissue at the pregnancy week of 6 to 8 weeks have different parts. The first part, the red one here, is the genital eminence, or what's called the genital tubercle. This is just to show you how external genitalia of the male and female are also descended from the same bipotential tissue during the process of embryonic or fetal development. That means the genital tubercle which is found here, when it is a male fetus, it will lead to the development of the penis. And when it is a female fetus, then this will form the so-called the clitoris. So the genital tubercle can be in the male, the penis part, and in the female, it is going to the clitoris. So this shows that also the different parts which are here, the urethral fold is giving rise to this penis shaft, and also the inner side of the external genitalia of the vagina, and the other outer part of the vagina are also originate from the genital fold here, and which is also the scrotum in case of the male. So the scrotum is a sac-like structure, which is a two-part, and it contains the testis inside it when the testis is descend from the abdomen. So this is just to show you that not only the gonads are originating from undifferentiated bipotential tissues, but also the external genitalia of the male and female are also originating from the different parts of, from the same bipotential fetal tissue. So in the development of the male external genitalia, there are two types of hormones which are important. One is testosterone. As I said before, testosterone, which is produced by the Leydig cells in the testis, is the one which is giving. So here we see the different parts of the male internal and external genitalia. That the darker ones are the ones which are testosterone dependent. That means the development of the vesicle, the seminal vesicle, as I said before, and the development of the vas deferens and the whole of the epididymis are all dependent on the testosterone. The other parts of the external genitalia, that is the scrotum here, and the penis, some part of the penis, and the prostate gland, they have what we call an enzyme called 5-alpha reductase in their cytoplasm. What they do is that they just change the testosterone to another highly potent form of testosterone, androgen. It is called dihydrotestosterone. So this is also an androgen, but it is highly potent, and it can bind to androgen receptor in more potent than the testosterone. And through the transformation of the testosterone to dihydrotestosterone through the enzyme called 5-alpha reductase, then the development of the different tissues. Here we see here the scrotum, the penis, and the prostate glands are possible. So this is the way how external genitalia and internal genitalia are developed in the male, depending on the ability of the cells to so let me give you some very brief summary of the whole system, what I talked before. As I said here, the XY chromosomes combination, in the XY chromosome combination, we have the presence of SRY gene on the Y chromosomes. This will lead to the development of the primordial gonads to the testis. And in the testis, we have the Sertoli cells and the Leydig cells. The Sertoli cells are producing the anti-Mullerian inhibiting substance, that means anti-Mullerian hormone, which is leading the regression of the Mullerian duct. And in the Leydig cells, testosterone will be produced. The testosterone will lead to the Wolffian duct to transform to the epididymis, vas deferens, seminal vesicle, and ejaculatory duct. So through the action of the 5-alpha reductase, the testosterone will be changed to dihydrotestosterone, and this dihydrotestosterone is important for the development of male external genitalia, the penis, the scrotum, and the prostate, which are very important. This is just shows the very summary of the development which I just described before. In the female, again, we have the XX chromosomes. There will be no SRY gene because of the absence of Y chromosomes. And in this case, the primordial gonads will differentiate into the fetal ovaries. In the fetal ovaries, because of the absence of the Mullerian inhibitory substance or the anti-Mullerian hormone, the Mullerian ducts will develop into uterus, the fallopian tube, or what we call oviduct, or the inner side of the vagina. Because of the absence of testosterone in the fetal ovaries, the Wolffian duct will no longer be there. It will regress, and then development of the outer vagina and the female external genitalia will be possible. So this is, I think, a kind of summary of the ones which I did yesterday, so that it will be a little bit more clearer, and a summary of the things which I did. So maybe in the next one, today again, we will continue on. Tomorrow we will continue on showing how the different reproductive systems are controlled at the central nervous system, in the hypothalamus, antipituitary, and the gonadal system, how the hypothalamus, anterior pituitary, and gonadal system works. And I think that is more clearer than yesterday. And these are the very important, important points what I want to point out from yesterday. So if you read, I don't know, everybody have access to the ebook. In the ebook, there is a text which is very nicely written, and that explains all the things which I have here. And then you can read on that one, and then if necessary, I will revise some of them in the final dates of our lecture next week. Yeah, I think that's all. Abby can stay in the chat for you to answer questions. I've seen some of the chats yesterday. She has responded very well, and then that's all for today. And we see each other tomorrow. Thank you.